Hello everyone, I am Shurasri Pal. I am a Prime Minister's Research Fellow, and we are discussing this question from GATE Examination 2022, Paper Life Sciences, question number 90. The question states, which of the following is are responsible for reversible receptor-ligand interaction? A. Ionic interactions, B. Hydrogen bonding, C. Peptide bonding, and D. Hydrophobic interactions. This question is MSQ type, so there can be multiple answers. Now, a receptor and a ligand, this reaction is usually, or this interaction is usually reversible. So it means that the receptor ligand interaction usually is transient, and the ligand binds to the receptor, moves away, binds to the receptor again, moves away. It kind of works in this motion. So no receptor ligand interaction is usually tight, or it's not a covalent interaction. So receptor and ligand, they can bind to each other and they can dissociate. So they so usually that is why they have an association constant and as well as a dissociation constant, which determines what is the affinity of their binding and uh, how much tightly they bind to each other. Now, molecules, they have an innate affinity for one another due to various types of interactions, like the electrostatic forces, like the Coulombic interaction, so the interaction between a positive and a negative charge, hydrogen bonds, and dispersion forces. The non-covalent interactions that result from this affinity are of particular importance in biological processes, including the catalysis of chemical reactions by enzymes. So it's talking about the interaction of the enzyme and the substrate, or the enzyme and the inhibitor, neutralization of foreign toxins by antibodies. So the antibodies that can bind to the toxins and then neutralize the toxins, that also depend on the molecular interaction, and also stimulation of cellular activities by hormones. So hormones, we know that they bind to their receptors. And on the cell surface, there are multiple receptors, and they have their particular ligands to which they bind, and then they can stimulate downstream signaling processes. So the cell signaling solely depend on this kind of receptor ligand interactions. Like, for example, in the neurons, in the um, synapses region, we know that there are these acetylcholine molecules that bind to the acetylcholine receptors, which can initiate um, the transmission of the nerve signal, right? So these are several of these are the interactions that are important in the biological processes. And to initiate all these processes, receptors and ligands exchange interactions with solvent and solute molecules for interactions with each other. So the receptors and ligands, they also interact with the solvents. So they have to exchange these interactions with the solvent and the solute to interact with each other. So therefore, these non-covalent interactions, these can be ionic interactions, these can be hydrogen bonding, and these can also be hydrophobic interactions. So the hydrophobic interactions occur between two hydrophobic regions of the two protein molecules. Hydrogen bonding occurs between, we know that the electronegative atoms and hydrogen, and ionic interactions are um, the, the electrostatic interactions. But C, peptide bonding, this option is not correct because peptide bonding is a covalent interaction and it forms between two amino acids. But when a receptor and a ligand, when they interact, there is no such covalent interaction that takes place. So therefore, our answers here will be A, B, and D. Ionic interactions, hydrogen bonding, and hydrophobic interactions.